Hi, this is Ayesha. I am MPhil student from Cohort University of Science and Technology, KPK, Pakistan. I am here to present my research topic. My research topic is molecular detection of macrolide resistance gene in Staphylococcus aureus isolated from fish market. My presentation will include introduction, objectives, material and method, result, conclusion, and finally the recommendation. Staphylococcus aureus is one of the most common bacteria which is found in animals and in humans. But this bacteria sometimes become pathogenic and cause many infections. These infections are treated with antibiotics. Macrolide is also a broad spectrum antibiotic which is used against these infections. It's include erythromycin, clarithromycin, azithromycin, and etc. Some other drugs. But due to their constant and frequently used in treating different humans and veterinary diseases related with the S. aureus, it can cause an emerging case of antibiotic resistance in S. aureus and can cause a threat to the surrounding environment and the contaminated food and the consumer which is consuming it. So it is important to have systematic data about antibiotic resistance in different bacteria, so we can make important strategies to control the transmission of resistant gene to humans or to our environment. The objective of my research is to determine macro-lead resistant susceptibility pattern in isolated S. aureus from the market fish, to categorize the macro-lead resistance gene RMA and RMC in S. aureus. The methodology is started with collection of fish samples from different fish markets and then isolation of bacteria S. aureus by growing it on mannitol, salt, agar, and some other important biochemical tests to confirm its presence. After that, antibiotic susceptibility test was be done. So we in which can see easily that which strain of bacteria are resistant or which are not. After the antibiotic susceptibility test, we done DNA extraction of the resistant bacteria or resistant isolates which we get from the fish samples. After that, we run PCR for the amplification of resistant gene, and the result was then sequenced and statistically analyzed. The result of my research is that there are two important genes, resistant genes, which I was looking in as resistant isolates. First one is RMA and second is RMC. As we can see there, the percentage of RMC is relatively higher than the RMA, which is 48.7. So it is can be concluded that RMC is the most prevalent type of resistant gene found in macrolide resistant bacterial strain S. aureus. The conclusion is that the high prevalence of this resistance, which is a macrolide resistance found in S. aureus isolate, can be threat to the consumer, which are consuming these fishes, or the humans that are handling them, the fisherman or the market, the seller, the shopkeepers, or the person who is handling them and who is cooking them, and it can also transmit resistant gene to humans. So it is recommended that there should be a continuous monitoring and surveillance on the antimicrobial resistance pattern in organisms, especially microorganisms, so that we can control the spreading of these resistant genes to other animals and humans, because it's make the treatment of some other infection and critical diseases more complicated.